All right, good morning, everyone. So today we're going to continue from our discussion of the last lecture. So let me go ahead and start with that presentation. So we were discussing the importance of the protein serum transferrin in terms of not just binding iron, but regulating its solubility and also its cellular uptake. And so we talked about two different routes by which this protein may potentially be able to ultimately deliver the iron into the cytoplasm. And so we talked about one pathway that involved the use of a chelator once inside the endosome. And the other pathway we discussed was this one, where we talked about the possibility that maybe the iron directly coordinated to transferrin in interaction with the receptor might be reduced to iron 2, and that reduction is enough for that metal to lose its affinity or its significant affinity to the protein and then be released. Released and then delivered outside of the divalent metal transporter. So while transferrin is one of the most studied proteins, this actual pathway of how that metal is ultimately released, there's still not 100% agreement. And so what I wanted to briefly talk about was some of the insight that was published last year by the students of the Metals of Medicine course, where they explored the possibility of serum transferrin playing a role in terms of the uptake, transport of non-ferric metal. And so these were some of the models or modes of possible entry into the cells. So one being that perhaps the metals may canonically bind to the protein. So by canonically meaning bind exactly like iron is able to bind the protein, induce a conformational change, and then be recognized by the receptor. But there's also suggestions that perhaps you don't necessarily have to follow this modality, and instead you can bind in an open conformation, and that enables enough of a protein change to be recognized by the receptor. There's also the suggestion that perhaps certain metals, have to utilize a mixed metallation form. And what that means is that iron is bound at one of the sites, and so that triggers a conformational change of the protein, thus enabling another metal to be able to bind to the protein, not necessarily in a closed conformation, but because of the iron presence there, that facilitates the uptake of the non-ferric metal. And then there's also the possibility of taking advantage of an advantageous surface site. And what that means is just taking advantage of any pocket on the surface of the protein, because there's already iron bound to the protein. And so you have a non-ferric metal that can just stick to the protein, and then because it is recognized and endocytosed, it is able to enter. Now, how the metal, the non-ferric metal, is ultimately released into the cytoplasm, well, there's some literature to suggest a couple of pathways. And so here, this is specific to non-redox active hard Lewis acids. And the reason for this specificity is because, remember, transferrin has the capacity to bind with greatest affinity for hard Lewis acid. So you can think about the transferrin metal binding site as a Lewis base, a hard Lewis base. And so one pathway is using acidification, chelation, and then the divalent metal transporter, or acidification, chelation, and then transport via ionophoric pathway. So let's visualize this. And so basically we're looking at this step where there is a possibility that you have the metal transformed into a divalent form, and that allows it to be delivered out. And so you're using there the divalent metal transporter. Or there's the possibility that perhaps you don't really need to alter the oxidation state of the metal, and there may be some biological chelator that can bind that metal and then transport it. So basically playing the role of an ionophore. All right, so these are possibilities. And as I mentioned before, they're not necessarily what truly happens within the human body. All right, so then returning to discussions about iron, this is where I ended in my lecture last time, where after iron is released from the endosome, you form what is known as the labile iron pool. And that labile iron pool is predominantly iron 2. Okay, so you have a really high amount of iron 2. Okay, but it does exist in equilibrium with some amount of iron 3, but again, more iron 2. And then that metal will get trafficked. And so by trafficking, what I mean is that some part of that metal will get stored into a protein called ferritin, and some part of that metal is going to get delivered to important biomolecules that is going to enable their functionality. And so what we discussed was this idea of iron 2 getting shuttled into the mitochondrion, acquired by ferrochelatase, and inserted into the protoporphyrin 9 moiety. And so that is what's represented here. Okay, that essentially is what forms heme. Okay, so that's the iron-bound protoporphyrin form. And as Manoj explained last time, you can have different varieties of this particular group, right, depending on the substituents that are available. Then heme, some part of it, can be transported out of the mitochondrion and into the cytoplasm and become part of the protein hemoglobin. And so at the end, 75% of that iron that you have ingested into your body, or that constitutes that homeostatic control of the metal, well, 75% of it is found in the form of hemoglobin. So it's in our red blood cells. Okay, and that's what's really helping us to be able to breathe. Another component of the iron is delivered to and formed what are known as the iron sulfur cluster. And so here you have some representations of these. And so this is the 2 iron 2 sulfide, 3 iron 4 sulfide, and this is forming a what basically is the components of a cubane structure. You see that more clearly here for the 4 iron 4 sulfides. So these proteins are very important in terms of electron transport and also for redox regulated enzymes. Right? So iron does not necessarily have to be in the heme form to facilitate that electron transfer or even redox. So let's talk a little bit about the amount of that portion of the iron that's actually stored. And so this protein, I alluded to it a few times, ferritin is actually extremely important. So it's a huge protein found in plants, animals, bacteria. So here's just a basic overview, 24 polypeptides, so it's roughly 20 kilodalton each. So we're talking about 480 kilodalton. And so to think about this, it's essentially like a factory to produce nanoparticles. You're forming these iron nanoparticles. And what exactly it is iron oxide that's being generated. And you're basically taking advantage of the low solubility of iron 3 plus. All right, so what's happening is the iron 2 that's getting shuttled into the protein, well, that iron 2 is getting converted into iron 3. And as I already mentioned to you guys, iron 3 is very poorly soluble. So essentially then, what you're facilitating is the mineralization of iron oxide. And this is being facilitated by peroxidases that are present. So the net result of this is that you are significantly increasing the amount of iron in a very localized environment. So remember, as I talked to you about this idea of compartmentalization, well, here within the confines of the actual protein, you're generating millimolar amount of metal. Okay, and this I'm putting in quotations because it's not soluble metal. But if you're thinking about it with respect to the concentration of the protein, millimolar amount, when the actual soluble amount of iron that's present within the cell is micromolar amount. So you have these really concentrated pockets within the cellular environment that is highly iron metallated. So as you can imagine, deletion of the ferritin gene, right, that's what will produce the ferritin protein, is deadly, right? We need iron and we need to be able to store it. So returning back to this figure here, we're looking at what I had mentioned to you, this idea of a physiological response as it depends on an essential element. And so this range that defines an optimal amount of metal. And so how does the body help to regulate that? So we have to take a part in terms of that regulation. You know, you are well aware of being careful about your consumption of iron, because if you have too much, it can be quite dangerous. So, what does the body do to try to maintain this? So, when the iron level in our body falls below the optimal level, we mobilize ferritin to access our stored iron. Again, this idea of mobilization, right? Because as I mentioned to you, ferritin is basically consisting of iron oxide nanoparticles. And so, in that form, it's bio unavailable, but we have the capacity, the machinery to be able to solubilize and make that metal functional again, bioavailable. All right, so let's go back and think about this and exactly how is ferritin doing this? How is ferritin doing this? Because the thing is that we have to maintain this homeostasis of the amount of iron that's going in, the amount of iron that's being shuttled out. So let's think about some of the things that you guys are well aware of. Biochemistry, you all have been exposed to this idea of metabolism. And metabolism is essentially mobilization. And so in the concept of metabolism, you're talking about biopolymers that are being broken down into its building blocks, and even further down than that. So into monomers and then into metabolites. So for instance, proteins broken down to their amino acid forms. So you have proteases that will facilitate that. So these enzymes that will break up the amide bonds and then forming these amino acids. Polysaccharides being broken down by amylase to form the monosaccharides. Triacylglycerols being broken down to fatty acids by lipase. Nucleic acids being broken down by, for instance, DNAs, RNAs into nucleotides. All right, and that is, one layer of the metabolic process. And so, as you're all aware, within the cellular environment, you have this well-known pathway, the citric acid cycle, and there's other cycles, but the citric acid cycle within the mitochondrion that then will further process these monomeric species, essentially producing or converting, if you want to think about it like that, some of this chemical form into 90% of the energy used by aerobic human cells. The nucleotides, at least the catabolism of these species, doesn't really yield a significant amount of metabolic energy. All right, so essentially that is what we have available to take advantage of these building blocks. So, these polymers are really important. They all have their function, but we also need access to the building blocks. All right, so now from this perspective, then let's talk about two possible routes by which we can mobilize ferritin to gain access to iron whenever we're starting to get too low in the amount of iron that's in our body. Well, one proposed route is the digestion of ferritin using the lysosome. Okay, and so here, this is a space within the cellular environment that has a lot of proteases. So you're basically facilitating the acid hydrolysis of proteins to then get access to that metal. So here, then, you're using acidification to solubilize the metal. Okay, now this is a very interesting route. I would say the controversy of this route is that you'll have this sort of influx of protons, which could be bad within the cellular environment, right? Because you want to have a control of the pH. The cytoplasm is 7.4. You don't want to, abruptly disturb that pH control. And so while this is an interesting route, it may not necessarily be a very likely route. Another possibility is a more controlled route, which is a regulated unfolding opening of the ferritin pores. All right, so how is that possible? Well, let's imagine a signal being sent indicating that, well, we need more iron, right? So let's take advantage of what we have stored. So you have the signal, and then you basically have a control over the pores of ferritin. The signal is sent, there's a recognition, and then there's an opening. And so that opening then facilitates potential small molecule reducing agents. And so I've talked about this briefly. There's a presence of ascorbic acid, glutathione, these small molecules that are very important in terms of redox. And so they have the capacity to reduce iron 3 to iron 2. So they're able to convert that iron 3 that's in an insoluble form into a form now that enhances their solubility. That would be coupled with chelation to bind that metal and further enable solubilization. All right, so you have that combination of a redox chelation process to then bring that iron back into the cellular environment for it to be used. Okay, all right, so that talks about what happens when you're right at the edge of deficiency. Then what if you're at the edge of just having too much iron? Okay, so our body has intracellular mechanisms, or even you can also think about some extracellular mechanisms for dealing with a reasonable excess of metals. All right, I say reasonable because there's just a really fine line there. You have what is known as an intracellular chelation. So the biomolecules within the cellular environment have the capacity to bind metal. So that for instance, what forms the labile iron pole are chelators that can help to make that iron bound for a transient amount of time, but still able to be delivered to other things. You also have processes known as extrusion, where a transporter is activated to expel an excess metal from the cell. A metal is shuttled to a storage compartment to make it inert. And then you have a process known as exclusion, where a metal's route of entry is removed, inhibited. All right, but as you can imagine, none of these are necessarily specific to iron, and that is actually the reality. There is no specific physiological route for clearing iron from the body. Okay? That is why it's very important that we participate in that control of how much iron we intake into our body. You know, if there's any route for release of the metal, it would be scratching your skin, basically shedding your skin. Okay? And then for women, women have the menstrual cycle, which is another route by which iron can be depleted. But what happens when we do have excessive iron that goes beyond just that minimal amount that's above what's needed? Well, let's go back to some parameters that I talked about before. I mentioned to you that iron that's delivered into the bloodstream, well, that's a very important process in terms of regulating that metal, solubilizing and controlling its delivery and biodistribution. And something that I didn't emphasize too much was that protein helps to bind basically the virtually 100% of that metal within the bloodstream as iron 3+. But it does not get saturated. So you have a distribution of this sort where you have about 27% saturated serum transferrin, about 34% mono iron serum transferrin. So that means transferrin that has iron at one site. You have 40% of serum transferrin that doesn't have iron bound. And I've labeled it here as apo. You know, there's a possibility that perhaps there could be other metals that are in those sites. You know, we're basically excluding that possibility here. But when you do have too much, so really talking about high levels of that iron, okay, well, the following can happen. Well, then you can saturate serum transferrin, and that's bad because now then the excess from that point on will then start to constitute the non-transferrin bound iron. And I had mentioned to you previously that there is possible small amount of that can form briefly when iron is released in the bloodstream. But truly, this pool of iron that's in the bloodstream is not necessarily a good pool. So this would constitute the labile iron pool within the extracellular environment within the bloodstream. So then what happens if you have too much iron? So you have the iron overload diseases. So toxic levels of iron can be achieved through the excessive intake in food, also multiple blood transfusions, okay? Mutation of proteins responsible for iron uptake and transport. That's something obviously you can't control necessarily, right? And so treatment efforts, we have efforts that are in terms of chelation therapy. Some older methods were bleeding patients. So you probably have seen, heard of stories or seen movies that, you know, that people being treated by bleeding them. And there's actually some therapeutic component to that, could be attributed to this. So here, just an overview of iron-related diseases. So the World Health Organization considers iron deficiency to be the #1 nutritional disorder worldwide, with about 80% of diets iron deficient. You have iron-related diseases, sickle cell anemia, thalassemia, iron overload, the hemochromatosis, so 1 in 250 persons of Northern European descent have this, acquired iron overload, and that's from multiple transfusions. Iron is implicated in a wide variety of other diseases, heart and liver disease, diabetes, neurodegenerative diseases, cancer, and restless leg syndrome. So there's a lot in which iron can actually be quite detrimental to our survival. All right, so that I wanted to emphasize this so you get that overview perspective of what is important about iron, but how it also can be quite harmful. Because I often hear these misconceptions or read these misconceptions in the literature like, oh, well, we can just use an essential metal because it's not toxic to us. But that idea is actually, a very controversial one because everything in excess or even too little, depending on if it's essential, can be quite dangerous to us. So this idea when someone tries to sell you a product for you to ingest because, hey, we need it to survive, so it's not toxic, that's a very controversial one. All right, so with this in mind, then let's move on to this idea about regulating metal biological speciation, as it goes hand in hand with this idea about transporting different metals. And so here, I'm going to take a few steps back so that we can, go over some of the basic coordination chemistry, but as it relates to biological speciation, we're going to actually formally define what that actually is. All right. So,
Professor,
yes, go ahead.
I have a question about like the previous PowerPoint. Because I do know there are conditions where you lack the ferritin supplies. So what happens in that case? Like, or you have very, very low ferritin supplies.
Right. So then there you would need to have supplements. So that's a way of just basically controlling the levels of the metal to be able to use it. So ferritin is a storage of the metal, right? It's absolute depletion. You would not be able to survive that. But to have a certain amount and then to supplement in a very careful manner, that's how you would be able to endure, because the idea is that you want to release the iron that's now going to get utilized, transported where it's needed. Right? So that's how you would actually control that. That's a very good question. All right, so a few things just to highlight about within coordination chemistry, but it actually relates to this idea of designing metal-based compounds that may have an application within the body, whether it's a therapeutic or maybe even for diagnostic purposes. This idea of like, what are those factors that are going to control a coordination number? And when we're talking about coordination number, really, whether it's going to be a low coordination number or a high coordination number. So this component of what's a low coordination number, let's say a coordination number less than 4. So this goes back into the theory of hard, soft, acid-base theory. And to enable this type of chemistry, it is favorable with a soft metal, soft ligand interaction, because now you have these polarizable species that can overcome the lack of a high coordination number. Because one of the fundamental things about coordination chemistry is that the bonds help to stabilize these compounds. So the higher number of bonds is good up to a point. Then something else that can help to enable a low coordination number is the use of large bulky ligands. And then if you're talking about a compound that has a charge, okay, so in this context, for cationic metal compounds, then you want to use anions that are of low basicity. And so why is that? Well, you want these anions that are going to help to balance their charge, right? But you don't want the anions to compete with the actual binding to the metal. So if you think about it, this context here where you have a charged cationic metal complex, right? And you need to have charge balance, so you have this anion that's going to facilitate that charge balance. So you get this net neutral compound, but you need to make sure that that anion does not have an affinity for the metal. So for instance, there are what are called these least coordinating anions that have a negative charge, but really does not have an accessible site for direct coordination to metal. So this is an example, which is humorously labeled as BARF. Now, on the other end, right, what's going to control high coordination numbers All right, so coordination number going greater than 6, well, that's going to be favored by chemistry involving hard metals, hard ligand interactions. And then what is part of that is the use of small ligands, right, to try to maximize coordination to the metal center. And then when we're thinking about a charged metal compound, well, here in the context of an anionic metal compound, so it's represented in this form, well, you want to use cations that have low acidity so that they serve as counterions excuse me, I forgot to write here, ligand binders. I will correct that. All right. Basically, you don't want them to compete for coordination to the ligands. So here is the representation of your cation that's going to provide the net neutral charge, so that charge balance, but it doesn't actually compete with the metal to bind the ligand that is there. So it doesn't exchange. Now the reality is, and I've mentioned this a few times, but it's important to reiterate that when we're talking about metal complexes, biomolecular ones in a biological system, you're going to have compounds that are, not exactly ideal. So you'll have a lot of distorted geometries due to non-homogeneous ligands. You're going to have constraints in bond lengths and angles. These are things that would be expected. All right, but let's talk a bit about what is regulating metals in solution. And let's start off from as simple as just thinking about the solvent. And when we're talking about biology, we're mostly talking about aqueous chemistry here. As I mentioned to you in the very first lecture, metal ions are never free. They are in fact solvent bound, and we were thinking about them in water. So here's just a representation of a metal that is aquated. So these water molecules are serving as ligands. When we're thinking about a ligand now that's not water binding to a metal in an aqueous environment, then now we have to think about it as a competition with solvent binding. So here we have this equation that represents this competition. You have that equated metal species that comes into contact with a ligand, and now that ligand has to have sufficient affinity for that metal to then outcompete at least one of the water molecules to be able to bind it. And so then here we have our representation of what is the formation constant for this metal ligand species. And one thing I want you to note in my representation here is that we do not take the water into consideration, because remember, your water is in excess here. So in terms of the thermodynamics, we're not factoring that in. So in the back of your head, know that the water molecules will play a role in terms of coordinating the metal as long as its coordination sites are not fully saturated by whatever ligand is binding it. All right, so just keep that in mind. But now let's talk about this idea of a formation constant, because I showed you the build-up to this idea of a ligand now outcompeting water molecules bound to the metal. Well, formation constants are a measure of the strength of ligand binding, and they also are a measure of the stability of a metal ligand complex. And there are two types of formation constant, the single step and the commutative step formation constant. So this is something you will learn in inorganic chemistry, and it's something I like to review because in the literature, you come across both representations of these formation constants. Now you have one that is single step. So what that implies is that you have a single metal ligand interaction. And what I will caution you is that we are now well aware that ligands, they have a denticity, so they can possibly bind on more than one site. Well, this representation of a formation constant is, I would say, an oversimplification, because we don't take in consideration the denticity necessarily. We simply consider one ligand, one metal binding, and giving you now this metal ligand adduct. And so your equilibrium constant K, your formation constant K, is a measure of the amount of that product versus the amount of your starting metal, your starting ligand. But the key thing being that the higher that value, that formation constant is, the more stable that complex is in the solution. Now, we can start thinking about, well, what if more than one ligand has to bind to the metal, okay? And so now when you're thinking about more than one ligand, there's going to be more than one step of binding. So here, I show you again that first ligand binding, so that's considered one step. But then if we consider a second ligand, well, we can think of it like this. We have generated a metal ligand complex. Now a second ligand is going to come in and interact with that metal center. And so you see that represented here. You have this other ligand coming in, and you form this adduct with one metal and two ligands. And so now we have to represent these two distinct steps. The first step is represented by K1. The second step is represented by K2. All right, so here then the expression or the use of the label K is for single step processes. All right, so that's simple enough. But what if we want to consider the possibility that the coordination itself doesn't actually go this way? That you don't have just one ligand binding and then the other ligand is just waiting for that binding to happen and then it has to come in. But what if they want to just both happen at the same time? You know, that's a possibility. So then let's consider that. Cumulative steps. All right, so here then, when we're considering this as cumulative steps, well, in the event that there's only one ligand available, right, that one ligand binding process, basically your K1 is what is known as a beta 1. Beta represents this idea of cumulative processes, and that's not so easy to see when you're thinking about just one ligand available. But now when you have two or more ligand available, here beta starts to make more sense, more practical sense. Again, this idea that you might have binding simultaneously. And so this represents that concept that you can have at least these two ligands binding virtually at the same time. And so to represent that then, we're going to use this beta 2 expression, which is the concentration of that metal with two ligand adduct divided by the concentration of the metal times the concentration of ligand squared. All right, so here then, beta really is a descriptor of a more than one step process. So you can consider this multi-step. And there's a way that you can generalize this expression. All right, so before I actually show you that generalized expression, beta 2 is actually equal to the product of K1 times K2. So if we were to work out the math, you will see that they're equal to one another. All right, so here's the generalized representation of how you can determine a beta expression depending on a number of different ligands coordinating to a metal center to give you a metal ligand adduct with not necessarily a defined number of ligands bound to the metal. All right, so now that we have distinguished between this idea of a single step and a multi-step or cumulative step process, well, now let's go back into this discussion of what are those factors that are influencing formation constants. And so one of the ones that we have touched upon a few times now is this idea of the hard, soft, acid, base theory, this idea of preferences, hard acids preferred binding to hard bases, intermediate acids preferred binding to intermediate bases, soft acids preferred binding to soft bases. And you can see that represented here in this table. And we actually discussed this without using these actual K expressions. We discussed this a couple of lectures ago, where everyone determined which type of ligand would have a preference for one type of metal versus another. All right, so you guys are well aware of this. What about other components that can influence formation constants? All right, so we did talk about the crystal field theory. So that's something that also takes part. All right, I don't have it in my list here, but we already know in our collection of knowledge now that also is a really important thing. Structurally then, you have what is known as the chelate effect. So the chelate effect occurs when coordinating through the same type of atom. A chelating ligand will outcompete a monodentate ligand for metal binding. And so here I represent to you chelation, in which you have chelators that can form 5 and 6 membered rings. And I emphasize these, right? So here's a representation of a 5-member ring, which includes the metal. Okay? Here's a representation of a 6-member ring, which includes a metal, these X's are atoms that are coordinating to the metal center. All right, so I'm emphasizing 5 and 6-member rings because it is this form of the chelators that provides the greatest stability. When you have chelators that form smaller rings, well, they often tend to have strain, and that strain has an energetic penalty. When you have chelators that form potentially larger rings, what happens then is that there's quite a bit of energy penalty. Because of that, you would have sort of an unfavorable ligand distortion to be able to try to coordinate onto the metal center, right? So preferentially, when we're talking about the chelate effect, we're talking about these 5, 6-membered rings. All right, but what about them is exactly enabling this enhanced stability? Well, in part, I've already mentioned to you guys that having greater number of bonds equals to greater stability. So a chelator will obviously have more bond interactions with a metal than a monodentate ligand, right? So that's a component there. When you look at factors that can really influence the thermodynamics, right? Gibbs free energy. So you have the component of the enthalpy and the component of the entropy. These play a big role. So enthalpy, just to remind us, involves bond breaking, bond forming. Entropy, this tendency towards disorder. All right, so we're going to look at that as it can play a role in terms of the influence on formation. So we have here a monodentate amine, which is methylamine, versus this bidentate version of that, which is ethyldiamine. Right, so you have 2 of these groups available to interact with the metal center. So in terms of the type of ligands they're identical, right? They have this nitrogen group with free pair of electrons that can facilitate coordination. So here, we're going to look at this idea of the monodentate ligand interacting with the with a metal. So I'm using cadmium as our point of reference. So just to give us some perspective, your cadmium here with your water molecules, we're thinking about this in an aqueous environment. You have your amine group that can coordinate to your cadmium to say that we have all 4 equivalents able to coordinate to the metal center, thus displacing 4 molecules. Well, you have this, right? These 4 methylamines coordinated to the metal center via the nitrogens. And then you have a K value of 3.3 times 10 to the 6. Technically speaking, that's a beta value. All right, but then you have the bidentate version of that same type of molecule. Here we have our perspective here to see how the same type of coordination is occurring. You have the same type of bonds forming, right? A nitrogen cadmium bonds in this compound that is forming, all right? But interestingly, the binding constant is much, much bigger, okay? So you have several orders of magnitude bigger. And the reason for this, it boils down to simply a difference in the number of molecules in solution. Well, where you have in the first process, if you're counting how many molecules they have on the left side versus on the right side, you will see that reaction B, which involves the chelator, you actually end up with a net increase in molecules. And one of the things that can drive a favorable process is an increase in entropy. And it turns out that the chelator effect, or the chelate effect, is entropy driven. But there's also another effect that's related to the chelate effect, and this is what is known as a macrocyclic effect. Okay? And we actually see this macrocyclic effect when we talk about the porphyrins. All right, so this is the idea of completely surrounding the metal ion with a chelator that is cyclic. Okay, and so here we have representations of these type of chelators that are wrapping themselves around that metal center. You see the associated affinity constants, they're huge. And it turns out that the macrocyclic effect is driven by enthalpy. And so I emphasize this sort of family of ligands because what we're looking at here are the same type of bonding interactions. We have nitrogen copper bonds in all of these molecules. But as you can see, the affinity constants are not identical. Even though you have the same type of bonds, you have 4 of them in each of these molecules. And you see that even going from, let's say this one is A and this one is B, there is a difference, a quite dramatic difference in terms of affinity, and that facilitated by this closing off of that component of the molecule. All right. So this is having to do with that ability of the nitrogen to be able to have a much more favorable interaction with the copper to strengthen that. So that is going to be what's the enthalpy component. So I want to ask you guys to just think about why potentially then would this other species have the highest affinity, the highest beta value? What do you guys think?
Conjugation?
Well, conjugation, but they're all okay. So okay, explain that. Now I know what you mean. Go ahead, explain. What do you mean by conjugation?
The excess of electron density going into the amine groups that contribute to better bonding towards the
exactly right. So you're talking about the actual this thing about the molecule, that you have this this aromatic this conjugation system that now enables this core amount of electron density, facilitating this interaction. Okay, all right. So that's a really big component here. So you have this highly versatile tetraaza macrocyclic ligand that's going to form fairly stable complexes. And so here we have the copper compound. You have the component of the negative charge. So that is, as we know previously, anionic ligands have a preference for interacting with these charged metal ions, these cations. And then, as you mentioned, there is the conjugation system, this aromatic system. So that's facilitating this density of electrons. In addition to, you have this high rigidity, excuse me, and I guess you can call it planarity of this core structure here, that is now going to favor that interaction between that copper and those nitrogens. So you can probably imagine a sort of square planar type of interaction here, with the possibility that there is possible coordination in an axial position. So this is something that you would see in the heme groups, iron coordinated to those protoporphyrins, right? That's also, very much this type of structure, and that what really stabilizes that type of interaction. All right, so those are a number of the factors that will enable the, a favorable formation constant for metal ligand interactions. But there's a component that we need to really discuss, which is the solvent. We have to an extent discuss how solvent can contribute to a sort of competition. But added to that, and so we're in an aqueous environment, is a pH dependence. So often, when we discuss metal complexes and their structure, we focus specifically on solid state structures. However, solution state structures are more diverse due to the impact of pH and the stability of certain metal oxidation states. So what I mean by that is that you may have a certain solid state structure for a compound, but you put that compound into solution, what may be in solution may not be at all identical to what you had in solid form. And that has to do a lot with the solvent, but also the pH. So let's define then pH-dependent aqueous speciation. So that defines the distribution of metal species in water at different pH values under thermodynamic conditions. For metal complexes, the focus would be on the different metal ligand species that exist in water. All right, so what are one of the components that has an influence? So one of the components, as we know, is the binding of water to the metal center, okay? But built into that component is this process known as metal hydrolysis, and that is the metal being able to lyse the water molecule. And when we think about this in terms of a descriptor, that is the metal acting like a Bronston-Lowry acid. So in other words, the metal, because of its interaction with the water molecule, is resulting in the dissociation of a proton. And so here I provide you a list of how you can generally gauge which types of metals are going to have a greater Bronsted-Lowry acidity. Okay, so typically we're talking about transition metals, okay, with high oxidation states that are going to be more favorable towards this metal hydrolysis. So, for instance, iron 3 plus is quite prone to metal hydrolysis, okay, which is the reason why, as I mentioned to you, iron 3 plus in pH 7 is if let's say you wanted to use that in a bioavailable format, well, that's actually a bad approach because that's going to be induce hydrolysis and is going to induce precipitation. Because oftentimes hydrolysis then affects the solubility of the metal, and so that can affect its availability. Vanadium, for instance, vanadium 5, it's quite extensively hydrolysis prone. So oftentimes when you're talking about vanadium 5 species, you're talking about them as these dioxyvanadium species. All right, but let's dive into this further. All right, so Zomi, you had a question, increasing the amount of molecules. What was that question in reference to?
Oh, it wasn't a question. It was my attempt at an answer to a question that you asked
Okay.
previously.
Oh, sorry about that. No, that's okay. Fine. What was the question that I had asked? Sorry, I'm old, so I forget. What was my question?
Oh, it's the question that I don't know who it was, but someone in the class answered.
Okay. Oh, was it about the higher affinity?
I think so.
Yeah. So it could be, but chelation is in general increases the number of bonds, so that's definitely a factor. Macrocyclic has more to do with like the arrangement of a ligand as it can interact with a metal to try to increase, to maximize the bond strength essentially. All right. A key point that I want to elaborate on is the effect that metal ions can have on water molecules, but also on ligands. That is that metal ions can lower the effective pKw of water. They can lower the pKa of ligands. And in our last lecture, we even saw that metal ions can even lower the pKa of molecules that are not even directly coordinated to it. If you remember, the dilysine trigger was an example of a lysine that wasn't even iron bound to the iron transferrin, and still just being in proximity to that metal was enough to influence its pKa. So that's something really profound about what metals can do. All right. So there, in the literature, you can come across collection of what are known as the hydrolysis constants, and these are things that you have to take in consideration when you're thinking about a metal compound, because hydrolysis and then these subsequent metal hydrolysis can really have an impact in terms of the speciation of the metal. All right, so here what I'm going to present to you guys is a case study, and we're focusing on the aqueous speciation of vanadium, so vanadium 4, vanadium 5. I know at least one student in this class does vanadium work, Omar, he does this, so he can probably give a better lecture than me on this. So vanadium 4, so here I'm just giving you a profile of the actual speciation of the ion itself. Okay, so we're just literally looking at the ion in water at different pH values. And think about that. That's just the metal ion at different pH values, and you have all these different species that are forming. All right, so what we're going to do, we're going to talk about what's constituting this. All right, so one of the things is that, just the highlights, solutions of vanadium 4 at acidic pH are stable. Okay, so I probably should have paired this with this statement over here, because in its absence it doesn't make 100% sense. What I mean by that is that the oxidation state of the metal will remain constant, will remain stable under these conditions. But when you start to go into the pH of 4 and 10, there is a strong possibility that the oxidation state of the metal will change, and you might have a preference then of vanadium 5, okay, because of just the solution conditions. All right. From a pH of 3 to 5, okay, so now let's draw our eyes to here and here, this range. We have vanadium 4 species that are cationic. All right, so you have this 2+ species, 1+ species. But above a pH of 5, we have these anionic species, right? So here, here. And then if you look closely at the actual formulations that we have here, well, then you have a mixture of Mononuclear, by that, what I mean by mononuclear is just one vanadium, and dinuclear species, two vanadium. So, for instance, this molecule here is an example of a mononuclear vanadium compound, whereas this species right here is an example of a dinuclear species. Okay, so relatively speaking, even though there are a number of species that are generated in solution and you know, depends on the pH which one is going to predominate, this is a fairly simple speciation profile. Okay, simple. Because when you compare it to vanadium 5, well, vanadium 5 can be quite complex. It's quite complex because you have a lot of hydrolysis occurring. All right, so let's look at the highlights of this speciation profile. So vanadium 5. Solutions of vanadium 5 are stable over a wide pH range. So what I mean by that is that vanadium 5 will likely remain vanadium 5 throughout this whole range that we're looking at here. At a pH of 1, so we're talking about right here, you have cationic species. So here's just the representation of that. But above a pH of 1, okay, you have anionic species. I think every single one of them above pH 1 is anionic. Okay. Another very interesting thing is that although there are some mononuclear species, and I believe there are some dinuclear species, the majority of these species are multinuclear. Okay, so that's, I would say, more than 2 vanadiums. So it gets quite complicated. And, you know, part of that complication is that they tend to be quite insoluble. Another really interesting thing, this more pertains to the cellular environment, is that in the presence of reducing agents, such as glutathione, cysteine, ascorbic acid, vanadium 5 can be reduced to vanadium 4. Okay, so that is a possibility, but that requires the presence of reducing agents. All right, so let's talk about the overview of the things that are going to influence the metal ligand aqueous speciation, because what I just emphasized was what's influencing metal aqueous speciation in the absence of, you know, of a ligand that is not the water, that is not water molecule. Okay, but now we're thinking about an actual ligand that can bind the metal. So you have ligand density playing a factor. So at how many sites it can coordinate to a metal. You also have the competition of the ligand to bind the metal and also to bind protons, because many of the coordinating sites on the ligand are also pronation sites. So you can have species such as this one that I show you here, where you have a metal that is bound to a free pair of electrons, okay, and it's still proton bound. But there are also conditions in which, in order to coordinate to a metal, to a ligand effectively, a depronation step has to happen, and then that leads to the coordination of the metal. And then you have the liberation of the proton. In an instant like that, then a competition is relevant, and obviously this will all be pH dependent. You also have, as I've mentioned, metal-induced hydrolysis. And as part of metal-induced hydrolysis is the relative affinity that a metal can have for hydroxide ion. Then you have the concentration of metal and ligand. So the speciation profiles of these interactions will depend on their concentrations, but also their ratios. You know, what if you have a metal in excess of ligand? Well, what if you have a ligand in excess of metal, right? That's going to have a really big impact in terms of the profile that you get. And another really important one is the solubility, right? Because the profile that you see, the speciation profile, are for the soluble species. But you're going to also have the component of some of these are going to be low solubility, and so some amount of that metal will get lost due to this thermodynamic sink of precipitation. All right, and that's going to affect the availability of the metal. All right, so that's discussions of metal ligand interactions. But now let's complicate this further. Well, what about in vivo aqueous speciation, right? Because now you have a whole ballpark of different things that can interact with your metal besides water and besides a ligand of interest. So in a biological fluid, you will have competition with low molecular weight species. You'll also have competition with high molecular weight species. Okay? And then another really important factor is the pH-dependent oxidation state of the metal. Whether that metal remains in a given oxidation state, or maybe it might get converted to another, and that can complicate the speciation. So let's go look at another case study. And here, we're going to focus on vanadium-4, right? We already saw vanadium-4 in the absence of ligands, just in water, and how it can depend on pH. Well, we'll look at a few vanadium compounds with anti-diabetic properties, okay? In a later lecture, we will talk about what about these vanadium compounds, or some of these that might have an anti-diabetic property. But let's just focus on the ligands in these compounds. So here's just a collection of ligands, of which they are bidentate. And here I show you a representation of how these ligands potentially coordinate to the vanadium. Keep in mind that you see that component, remember vanadium, vanadium-4, vanadium-5, hydrolysis prone. So you're going to have this vanadyl group, this vanadium oxide group, that's going to be a significant component of the speciation. Right? So here I give you a representation of a bidentate coordination, two of the molecules forming a square pyramidal structure, coordination number 5. And that can be quite favorable for vanadium-4. All right, now let's think about these compounds. All right, so all of these can form these type of compounds, every single one of these ligands. Well, let's introduce biological ligands. So now let's imagine putting these molecules within the body and just a representation, because there are many more. Citric acid, that exists in our blood, lactic acid, phosphates, these are very important. They have metal binding capacity. We can't ignore them. And even though I don't have them listed here, we can even think about proteins that have metal binding capacity. You guys already know serum transferrin, it has metal binding capacity. That has an effect. So a study was done to look at these vanadium-4 compounds and how they will speciate in the presence of some common biological ligands. So to the right, we have a summary of that data, a lot of data that was collected. And this little plot, it looks very simple, but the collection of this is quite complex. All right, so what we're looking at here is what happens to these small molecules when you have them in the presence of these biological ligands plus the proteins. They're also biological ligands if you want to think of them like that. And so what we see from this collection of data, so what do we have here? These are the percentage of vanadium-4 species, or these vanadyl species. And then on the right, we have the different ligands. So that slide of the ligands that I showed you the visual of. And then what actual complexes form, at equilibrium. Okay, and then we see the different types of species here. So the summary of this is that we do have some amount of the intact vanadium-4 compounds. So some of these compounds can remain intact, but a large majority of them do not. Okay, so for instance, if you want to focus on this ligand here, is abbreviated HDP. If you look at it here, you have a majority of the intact compound, because it says the vanadyl-4 with the ligand, it constitutes virtually 60% of what's found at equilibrium. And then at about 25% or maybe 30%, you have vanadium that's bound to the transferrin protein. Okay, so there's some amount of the intact compound, but there's also dissociation and binding to something else. So that compound did not remain completely intact under these conditions. And we're talking about conditions that are a pH of 7.4. Okay, that's actually really important when we're thinking about where the speciation is occurring. But for that one compound, but some of the other compounds, for instance, if you look at everything else, right? Everything else, if you notice, everything else is virtually, serum transferrin bound. So it's like about 70%. So you have a significant amount of dissociation of the vanadium. All right, so there's a good amount of exclusive vanadium-4 bioligand speciation. And then there's some amount of what is mixed ligand vanadium-4. So what I mean by mixed ligand, what that means, for instance, just to give you guys a visual, well, you have one of the original ligands bound to that vanadium, and then perhaps one of the biological ligands here, lactate, bound to the vanadium. So this gives you what is called a heteroleptic compound, right? So that shows you the potential distribution of that vanadium. And this is just to give you sort of a snapshot of what can happen to these compounds. And that is only specific to the 7.4. All right, so that just give you, just to look at that a little bit in a bigger perspective, but that's just the overview. All right, so then let's review then. Why is this important? So many things that we already talked about. And from the perspective of toxicological, pharmacological perspective, if you're thinking about a metal for potential use in the body, well, you want to know some information. For instance, you want to know, how do biomolecules regulate a particular metal in vivo in terms of solubility, oxidation state, maybe even potential role for cellular transport, biodistribution, even excretion. Some metals are not easily eliminated from the body. As I mentioned to you, iron is not easily eliminated from the body. So these are really important to understand when you're thinking about something that you're going to, as far as a metal that you're going to put into your body. And then, well, you better understand whether it can even survive the presence of all of these other bioligands, because if it can't survive that, there's a very little chance that it will be able to reach its target. So that goes into this idea of a biological fate of a metal compound. Well, how can the body metabolize these compounds? Are there any harmful metabolites? Can it survive the route of administration? For instance, the pH of fluids. So I only talked about that case study at a pH 7.4. But let's say you want to design something that's orally digestible. Then you have to think about, well, can it survive the acidity of your stomach? Can it be able to be transported into the intestinal cells and into your bloodstream? Or what if not even there? What if you need to deliver it into your cerebral spinal fluid, right? These are things that you have to think about. Does it have the capacity to reach its target? Can it be cleared from the body, right? And if it can't be cleared from the body, well, what is its relative toxicity? Maybe it's okay that a certain amount is distributed and it won't harm us. These are really important factors to consider, and why speciation, although it can be quite complex, is important to gauge an understanding for. All right, so any questions about these topics? I know there's a lot of material, but these are materials that really helps us to think about the design of components that contain metal that we may need to use for an application in our bodies. Any questions? All right, so if no questions, well, have a great day, and I'll see you guys next time.